Assalamualaikum and good morning everyone. So today we continue our lesson with plant hormones. Okay, so these plant hormones apparently they are growth substances, right, that acts as chemical signals that bring responses that regulate growth and development in plants, right? So for STPM level, okay, there are 5 major classes of plant hormones that you need to know. They are the auxin, okay, do mind the spelling, gibberellins, right, double B and double L there with S at the end there. Then cytokinins, abscisic acid, and ethylene, right? So students, please mind the spelling, all right? A small or minor error on the spelling, you'll be penalized in the exam. All right, so the characteristics of these plant hormones, right? They are effective to regulate growth and development in extremely small concentration. So meaning to say these plant hormones, they are only needed in small concentration, okay, to regulate the growth and development in plants. And then produced by young cells at the shoot apices, root, ovary, embryo, seed, and injured cells. Now, these sites that produce these hormones, these plant hormones, it depends on the type of hormones, right? Different type of hormones have they are being produced or synthesized at different locations, but generally these are the locations where these plant hormones are synthesized, transported in plants by diffusion, right? And then these plant hormones act by signal transduction, right, which an external signal will bring about an internal signal that causes one or more responses in the cell. Plant hormones may respond to signals from other hormones. These bring interactions that enhance or antagonize each other. So we need to say plant hormones, they can work together or give antagonize effects on each other. Each group is related chemically and there is no chemical similarity. Okay, so now let's go to the first one, auxin. So apparently this is how an auxin look like. No, they are chemical structure, right? Okay, so auxin, they are plant hormone that exists in natural form, right, as indole acetic acid. So I need to say auxin, you can write as auxin or indole acetic acid, IAA. In the exam, please write the full name. If you choose auxin, okay, mine is an X here, not K. Okay, yeah, this is an X. It's not K with S. Okay, it's an X here. Normally, candidate they will write as AUKSIN. It's wrong. You can write as auxin or you can write as indole acetic acid. But if you choose to write as indole acetic acid, make sure the spelling is correct. Okay, and derivatives of auxin is naphthalene acetic acid. Okay, and its analogs, which is 2,4-dichlorophenoacetic acids. Okay, this is not that compulsory for you to know for your level, but indole acetic acid and auxin, you have to know these two names. This auxin produced in very dilute concentration in the tips of shoot and root apices, floral buds, young ovaries, embryos, and injured tissues. Okay. Now let's go into the characteristics of auxin. Okay, transported by diffusion and has direction. Okay, more sensitive to root cells than stem cells. If you look in this diagram here, this chart here, root cell respond, okay, better at low auxin concentration. You can see the auxin concentration here, okay, which is against the percentage of growth in the plant. Now in roots, you only need small concentration of auxin, right, that can stimulate or trigger, okay, the growth in the root cells. High concentration of auxin, however, it will inhibit, okay, the root growth. You can see here, as we increase the concentration of the auxin, it will inhibit the root growth. But higher concentration of auxin will stimulate, or high concentration of auxin will stimulate, okay, or increases the shoot's growth. Auxin moves laterally away from light. Okay, it doesn't like light. Since it moves laterally away from light, okay, this will causes shoot of plants to show positive phototropism, right? That's why the shoot of the plants, they show positive phototropism because of this auxin. It moves away from the light. Can be broken down by strong light. That explains why auxin dislikes sunlight. Okay, so meaning to say, when this shoot, it grows towards the sunlight, when the shoot shows positive phototropism, the parts of the shoot that is being exposed to the sunlight, okay, is lack of auxin. Okay, and parts of the shoot, okay, we are talking on the same shoot that has two parts that is exposed to the sunlight and parts that is not being exposed to the sunlight. That part, okay, has higher concentration of auxin. Okay, so can be broken down by strong light, pH, and IAA oxidase enzyme. Reactions of auxin can be inhibited by chemicals, right? So yeah, this is what I wanted to show you earlier. Okay, so you can see, okay, how this shoot can show positive phototropism. This is the part of the shoot, okay, there is lack of auxin. It grows toward the sunlight. Part of the shoot that has high or higher concentration of auxin is not being exposed to the sunlight. Right. Now, roles of auxin, okay, induces growth, okay, by cell expansion, right? Then, and this cell expansion can be explained by the acid growth hypothesis. For STPM, this acid growth hypothesis is not in your syllabus, so I'm not going to explain it here, right? And then auxin causes positive phototropism and positive geotropism, right? Which is something I didn't mention earlier. Positive geotropism by accumulating the dark side of a shoot tip that is exposed to a unidirectional light and at the direction of the gravity of a root. Okay, next, auxin stimulates and activates production of meristem cells for secondary thickening, and then inhibits development of lateral buds, okay, but enhances or promotes apical dominance. That's why trees, they grow, or plants, they grow tall, okay, because of the apical dominance, which is being promoted, okay, by auxin. You can see here, okay, this is the apical dominance, and this lateral buds here, the growth of these lateral buds is inhibited by the auxin. If we cut off or we remove this apical bud, what happens, like here, okay, the lateral buds will progress or develop into lateral branches, right? That's why if you want to make your tree bushy, your plants bushy, you cut off or you remove the apical dominance, or the apical bud, sorry. Okay, so the lateral buds will develop and develop into lateral branches. So that makes your plants or your trees bushy. Okay, auxin induces root initiation from cutting and calluses. So this is calluses. Okay, parts of the trees that when you chop off or you remove, it becomes something like this. All right, it hardens. So if you apply auxin here, okay, to these calluses, earlier when it was being chopped off, okay, this branch being chopped off, if you apply auxin here, it will induce growth of new cells, okay, but once it already formed callus like this and hardens, it won't. Promotes fruit growth and induce fruit development without fertilization, okay, or you can simply say that auxin induce parthenocarpy or promotes parthenocarpy, right? fruit development without fertilization. That is parthenocarpy. Stimulates stem elongation. Okay, this is only at low concentration of auxin. Promotes the formation of lateral and adventitious roots. Okay, auxin does not promote formation of lateral buds or development of lateral buds, but for roots, yes, it does. It promotes the formation of lateral and adventitious roots. Promotes vascular differentiation, okay, and inhibits leaf abscission. Okay, there's a diagram here. In your module, there's a diagram here, right? The leaf abscission. Okay, luru, down luru. So it prevents the leaves from fall. Okay, so leaves that fall, meaning to say it has very low or absent or lack of auxin. Okay, leaves that is still intact and green means that leaves has high concentration of auxin. Okay, next, gibberellins, double B. Okay, gibberellins. Okay, I think gibberellins, yes, single L, right? Double B and single L. But there's some books uses double L as well. Both can be accepted. Okay, gibberellins, they are plant hormones that exist as gibberellic acid. So you can write as gibberellins or gibberellic acid, but not Ga3. Produced in young organs such as apical leaves, developing buds, root tips, and germinating seeds, and transported by diffusion. Okay, this is the chemical structure of gibberellins, right? Now, roles of gibberellins. Okay, promote stem elongation and vegetative growth in plants. Okay, then break dormancy in seeds and buds. Or you can simply say that gibberellin stimulates seed germination. Promotes flowering in long day plants, right? Promotes fruiting and parthenocarpy in certain plants. Okay. Promotes apical dominance. So meaning to say gibberellins and auxin, they work together to promote apical dominance. Alright, so these are some of the exercises. Okay, you may pause the video now. Try on your own to do these exercises. Okay, after that you come back here. Okay, done. Alright, so let's go to the questions. How does gibberellins promote stem elongation and vegetative growth? So apparently gibberellins promote stem elongation and vegetative growth in plants with the presence of auxin, right? By activation of certain genes in plants. List out with examples the effects of gibberellins on stem elongation and vegetative growth. Okay, so the examples are dwarf plants. When treated with gibberellins, will have normal growth. Okay, and then gibberellin causes bolting in rosette plants such as cabbage, where the stem lengthens like ordinary plants. So I don't have the diagram here for this bolting. You may Google it up. Okay, bolting in rosette plants. It will direct you to images that show you this bolting. Explain how gibberellins break dormancy in seeds and buds. Right, as a response to the increase of the environment temperature, especially after winter, okay, gibberellins will synthesize hydrolyzed enzyme that hydrolyze the food stored in the seeds for germination. Okay, what do you think will happen if a long day plant has a longer night length, okay, longer dark period? I think I should rephrase that. What will happen if a long day plant has a longer dark period or has a dark period that exceeds the critical night length and treated with gibberellins? What do you think will happen to this long day plant? Okay, what happen is gibberellin will promote flowering in long day plants, although long day plants can only produce flower if exposed to longer dark periods. Sorry, not longer day length. Okay, this is wrong. Not longer day length. Only produce flower if exposed to dark period that is longer or exceeds its critical night period. or night length. Name another hormone that can enhance the effect of gibberellin on apical dominance. I did mention earlier, auxin. Okay. Oh, this is the bolting. Okay. All right. This is the rosette plant. Applied with gibberellin, it bolted. Okay, the shoot bolted. All right. Next, cytokinins. These are plant hormones that exist in natural form as zeatin. Okay. Produced in the embryo of fruit and seeds. Right. So every time when you eat the fruits, you're eating cytokinins. Roles of cytokinin. Okay, regulate cell division in shoots and roots by promote cell division and cell differentiation in meristems, injured cells, and callus with the presence of auxin or gibberellins. So cytokinin works together with auxin and gibberellins to regulate cell division in shoots and roots. Delay the process of senescence, especially in leaves. All right, senescence is aging. Okay. Inhibits apical dominance in shoots but promotes lateral bud growth. Inhibits formation of lateral roots. Okay, but promotes apical dominance in roots. Right, and this is together with the presence of auxin. Promote movement of nutrients into sink tissues and stimulate seed germination. Right, I'm sure until now you know what is sink tissues or sink organs. Okay, exercises on cytokinin. So you come back here after you have done these exercises. Okay, all right, done? Okay, that was quick. Name two other hormones that enhance the effects of cytokinins. So, auxin and gibberellins. How does cytokinin delay the process of senescence, especially in leaves? Okay, it retains the activities of the chlorophyll. Right, that's why dead leaves or fallen leaves, sorry, dead leaves, all right, they are brown, meaning to say that all the chlorophylls are dead. How does cytokinin promotes development of lateral buds into branches? Okay, by inhibits apical dominance. Explain how does cytokinin determines the formation of root and shoot in plants. Okay. Cytokinin inhibits the formation of lateral roots with the presence of auxin. Thus, works together with auxin to determine the formation of roots and shoots. Okay. All right. So we are going quite fast here. Okay, next is abscisic acid. Okay, please mind the spelling. Abscisic acid, synthesized in the chloroplasts of shoots, more concentrated in the leaves, fruits, and seeds, right? And promotes dormancy. Okay, so abscisic acid will inhibit growth in plants. Okay, so this abscisic acid apparently has, okay, antagonistic effect compared to auxin, gibberellins, and cytokinin. Okay, antagonistically means it's the opposite. Okay, the abscisic acid has left effect, auxin has right effect. Okay, it's the other way around. Okay, now roles of abscisic acid: it promotes abscission of leaf and fruits. This abscission is the aging process. Abscission of leaf will cause the leaf to fall. Right. Promotes dormancy in buds and seeds. Remember, gibberellin breaks seeds dormancy. But for abscisic acid, it promotes dormancy. Promotes closure of stomata under conditions of water stress. Okay, so these are the three roles of abscisic acid. Okay, exercises. All right, hopefully you have done. Okay, if you're done, then let's proceed. Explain what will happen in a plant, especially in the leaves and fruits, when the concentration of auxin drops. Abscisic acid, sorry, abscisic acid will promote abscission of leaf and fruits by inducing the formation of abscission layer as concentration of auxin drops. How does dormancy in buds and seeds due to abscisic acid can be inhibited? treatment with auxin, cytokinin, and gibberellin. Why? Because these three hormones, okay, has antagonistically effect compared with the abscisic acid, right? Explain how does abscisic acid promotes closure of stomata. Under conditions of water stress, the abscisic acid stimulates the loss of potassium ions from the gut cells. So what happens? Water potential in the gut cells increases, causing water to diffuse out from the gut cells into the subsidiary cells. So gut cells become flaccid, curve inward, and the stomata closes. All right. Ethylene. All right, the last gas, the last hormones in plants for our syllabus or level. Ethylene or ethene. Okay, these are hormones in the form of gas, right, that induces fruit ripening. All right. Ripening is single P, not double P. So produced in ripening fruits, nodes of stems, aging leaves, and flowers. Okay, the roles to promote ripening of fruits by inhibits the synthesis of DNA and protein in the fruits, promotes abscission of leaves by forming the abscission layer. So ethylene works together with the abscisic acid. Promotes etiolation, which is the yellowing of plants due to lack of lights, and inhibits growth of roots. Okay. Okay, so auxins, the transportation of auxin is downwards. Okay. Okay, while the transportation of cytokinins is upwards in plants. Okay. Auxin is favored by the rooting, while cytokinins is favored by the development of axillary buds. Okay, these are the summary roles of the plant hormones. Okay, this is taken from the Campbell textbook, if I'm not mistaken. Okay, right, either Campbell or Raven textbook. It's either one. All right, now the last one for today: application of these plant growth regulators. Okay, or plant hormones in agriculture. Okay, sorry, plant growth regulators, okay, means synthetic derivatives of plant hormones. Okay, so meaning to say the plant hormones, they are being produced in the lab to produce a synthetic plant hormones, and the synthetic plant hormones are known as plant growth regulators. So the first one we have auxin or synthetic auxin, right? Auxin or indole acetic acid. So this auxin or synthetic auxin used to induce formation of shoots and adventitious roots. Okay, this will increase the number of plants asexually. Okay, used to prevent abscission and fruit drops, thus induces normal fruit growth. And then used to promote parthenocarpy, so this will causes the fruits to be seedless, bigger, and maximum in numbers. And auxin, all right, in agriculture also used as herbicides, which selectively kills weed and inhibits sprouting. Okay. Now gibberellins, okay, or gibberellic acid. Again, when it comes to gibberellins or gibberellic acid, the B is a double B, all right? The L can be double or singular. Okay. So produced as gibberellic acid by fungal cultures, used to promote parthenocarpy, production of seedless fruits and larger fruits, similar with the auxin. Used to break seed dormancy and stimulate seed germination. Okay. Now ethylene. Okay. The type of ethylene that is widely used, the synthetic ethylene, is ethylene, right? And then used to promote ripening of fruits. Okay. Then used to control the ripening of harvested fruits. Okay, only three. Right. So this is how ethylene, or synthetic ethylene, is being used in the agriculture. So one example is like the farmers. Okay, they can pick up when they harvest the fruits. Okay, the fruits that is being harvested. Okay, we assume that these fruits are going to be exported. Okay, now when it comes to exports of fruits, it takes days to weeks. So if they export the harvest ripe fruits. When these fruits arrive at the certain country's destination, the fruits will be already rotten, right? So what happens? They will harvested non-ripened fruits, all right, buah mentah. So when these fruits reach, okay, the country destination, okay, the supplier, okay, they will spray these fruits with the synthetic ethylene. So the synthetic ethylene will promote, okay, the ripening of the fruits, or it may control the ripening of the harvested fruits. Depends. Abscisic acid or synthetic abscisic acid, right, used to promote wilting and senescence. Used to promote bud and seed dormancy, right, and used to inhibit plant growth. Okay, that's abscisic acid. Cytokinin, okay, used to delay aging and senescence of plants, all right, in the agriculture. Okay, I think only one. That's it. All right, so see you again in the next lesson. Okay, bye.